Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Please like, subscribe, and share with your friends and relatives. It really helps support the Sand Science Experience. Today, we're taking your deep dive into some really fascinating research you've shared with us. We're delving into something that, it used to be dismissed as genomic noise, but now it's looking like a potential key player in aging. Nuclear mitochondrial DNA transfers, or NUMTs for short.
It is quite a story, actually. Fundamentally, NUMTs are segments of DNA from mitochondria. That's the powerhouse of the cell, remember? That have somehow got themselves integrated right into our main nuclear genome.
Right.
And this isn't actually new, evolutionarily speaking. It's a very old process, a relic of that ancient symbiosis.
Okay, so an old process, but for a long time, people just sort of ignored them, thought they were just leftover bits, inert pseudogenes.
That's exactly right.
Oh.
Pretty much seen as baggage. But the papers you've brought in, they suggest our view has, completely flipped. They seem to be quite dynamic elements.
Like finding a hidden control switch you never knew was there.
Precisely. And we're now getting some compelling evidence that these NUMTs aren't just, sitting there passively. They might actually be able to tweak nuclear architecture, influence how cells function.
Wow. And what really gets me is this. With modern whole genome sequencing, we're not just seeing the ancient ones. It seems endosymbiosis, the actual creation of new NUMTs, is happening right now in us.
Yes, it appears to be an ongoing process, which is, it's quite something to think about. Our DNA might still be getting subtly rewritten.
So how does that happen? How does mitochondrial DNA actually end up in the nucleus?
Good question. The main mechanism seems to be linked to stress. When mitochondria get damaged, say by oxidative stress, they can release their DNA, their mtDNA.
Okay.
And this free mtDNA can then get integrated into the nuclear genome. Often when the cell is trying to repair breaks in its DNA, it uses a pathway called non-homologous end joining, NHEJ. It's like a quick fix repair system.
A quick fix that sometimes grabs the wrong piece of material, maybe?
Kind of, yeah. It patches the break, but might incorporate this mitochondrial fragment in the process.
And crucially for aging, this whole process, this integration, it seems to happen more in aging tissues.
That's what the evidence suggests, yes. The older the tissue, perhaps the more mitochondrial stress, the more DNA breaks, and potentially more NUMT integration.
Okay, so there's a direct link building up.
Exactly. And looking at the bigger picture, studies are showing correlations. More NUMTs accumulating in the brain seems to correlate with cognitive decline, reduced lifespan. We see this in human brain tissue, and similar increases in older rats' brains and livers.
Right. It's not just theoretical. There's that striking example with rapamycin treatment, too, isn't there, in aged mice?
Ah, yes, that's a compelling one. Treating aged mice with rapamycin seemed to reduce mitochondrial reactive oxygen species. That's a key stressor.
Mm-hmm.
And it also reduced the accumulation of NUMTs in their liver cell nuclei. It suggests a link, maybe even a target for intervention down the line.
So let's connect this to the hallmarks of aging. How do NMMTs fit in? Genomic instability seems like an obvious one.
It does. NUMTs have definitely been observed where there's genomic instability. For instance, age-related increases in NUMTs in mouse immune cells,
Mm-hmm.
T lymphocytes, they correlated positively with micronuclei formation.
Micronuclei. Those are little fragments of chromosomes left behind after cell division goes wrong, right? A clear sign of instability.
Precisely. Then there's epigenetic alterations. When an NUMT inserts itself, it can potentially mess with how DNA is packaged, the chromatin structure.
Uh-huh.
And it might even create new sites for DNA methylation, which could switch genes on or off inappropriately, affecting gene regulation as we age.
Okay, so influencing gene expression patterns. What about mitochondrial dysfunction itself?
Well, that's where it gets interesting, possibly a feedback loop. Stress damages mitochondria. They release mtDNA. This mtDNA floating in the cytosol acts as what we call a damage-associated molecular pattern.
Like an internal alarm signal.
Exactly. It triggers the cell's innate immune sensors, like the cGAS-STING pathway. This can promote inflammation, and chronic inflammation is, it's a major driver of aging.
Right, the inflammaging idea. And you mentioned telomeres, too. Is there a link there?
It's more speculative at this stage, perhaps less explored. But the idea is, if NEMTs happen to insert near telomeres or centromeres, critical chromosome structures, could that affect their stability or function?
Potentially influencing telomere shortening, maybe?
Potentially. It's an intriguing thought, but needs more research.
So the big question hanging over all this is, are these NUMTs just like passive markers, scars left by aging and mitochondrial problems.
Or do they actively drive some aspects of aging themselves?
Yeah.
By messing with the nuclear genome's structure and
Yeah.
function, that's the key thing we still need to figure out.
Right, cause versus correlation.
Exactly. The jury is still very much out on that.
But either way, even if they're just markers, their potential seems huge. As biomarkers for tracking aging, for assessing disease risk or progression.
Absolutely. They could give us a window into cellular health. And maybe, just maybe, if they do have an active role, they could become therapeutic targets themselves. New ways to potentially tackle age-related decline.
Fascinating stuff. Okay, that brings us towards the end of this deep dive. Please remember to like this video, subscribe to our channel for more deep dives into cutting-edge science, and do share it with your friends and relatives. It really helps support the Sun Science Experience. This has been another deep dive from the Sun Science Experience.